Could you possibly talk a little bit about why you study Salmonella enterica and why it's interesting or important for public health or whatever to learn more about its infection mechanism?
Salmonella is a very important pathogen, both in the United States or the industrialized world, if you will. It's Europe and the United States. It's probably the most important cause of newborn illness in these countries, in the US and Europe. And so it's a very important pathogen that causes more than 1 million people probably get infected with salmonella. But in addition, in developing countries, salmonella also is a very serious problem, and probably a couple of 100,000 people die of salmonella infection every year. So it is a very significant public health concern, both in the industrialized world, but also, more importantly, in the developing world as well.
Okay. And then you're specifically studying the type 3 secretion system?
Well, our interest is to understand how salmonella causes disease. That's sort of the main interest. And we have different angles or different aspects of the mechanisms by which salmonella causes disease that we study in the laboratory. But one of the most important aspects is the study of this remarkable device that salmonella has that is central for its ability to cause disease. And this device, for all intents and purposes, you can sort of envision it as a tiny little syringe, that is a nano syringe, that has the remarkable ability to inject bacterial proteins into mammalian cells, into the cells of the body, for example, cells that cover the intestinal epithelium. When salmonella is usually acquired through the oral route, either by contaminated food or water, and then once it encounters the intestinal epithelium, it injects a battery of bacterial proteins that those proteins have the ability to coax the cell to do things for salmonella. So in a way, salmonella commandeers a lot of cellular functions for the benefits of the pathogen. And among the things that the bacterium makes the cell do is to make the cell to take up salmonella into the inside of the cell. So it's not a forced entry, but rather is fooling the cell into doing things so that the bacteria will end up inside the cell. But the whole manipulation, if you will, is done through these proteins that have different enzymatic activities that allow bacteria in a very complex way to manipulate many different aspects of the workings of the cell. So that this nanosyringe is so central to basically the ability of salmonella to cause disease that, of course, we are very much interested in understanding how it works. So that's basically the background, if you will, for one of the things that the laboratory does.
Could you briefly explain just what we already know about how it works and what we don't know about how the...
Well, we know that these nanos, we are able to isolate this syringe. In fact, you can see a nice picture in there. So we can isolate it. We can study what are the components of the machine, so that we know that. And we know certain things about how this machine selects the proteins that are destined to be injected. So now, more specifically, this sort of new discovery that we just recently made addresses a very important aspect of how this machine works. And one of the key elements for the machine, this nanomachine, to be able to do what it does is to be able to select the proteins that are going to be injected in a particular order. Okay, certain proteins need to go first, second, and third. So that ordering is essential for the workings of the machine. If somehow the ability of the machine to order the proteins, to put the proteins in certain order to be delivered, is altered, basically the machine loses the ability to do what it does.
Right.
So I suppose the main discovery in this recent piece of work is that we have sort of identified the mechanisms by which these proteins that are destined to be injected at order so that they can go in a particular fashion. So you can imagine that if you have a delivery system and certain things need to go first, second, and third, like if it was a sort of a UPS or a FedEx delivery track, there will be certain cargo that is going to be put at a sort of privileged position so that then when you are about to deliver, you open the door and you go to those first. So we have discovered that there is a platform, if you will, inside the bacteria that's the job of sorting these things. And that's a very important discovery because, as I said, that is central to the ability of the machine to work. So in basic science, we want to understand how things work. We don't necessarily, laboratories like ours are not engaged in research that is directly aimed at developing any kind of drug or pill or anything like that. That's not what we do. But we do generate the knowledge around which you can design, for example, drugs that will interfere with the process that we described. So, for example, this platform could be a good target for, say, pharmaceutical compounds that could inhibit the workings of this particular machine, and then therefore you could potentially disrupt the workings of the machine, and therefore you could potentially have, say, a drug that will interfere with the ability of Salmonella to cause disease.
And would these types of antibiotics be better in some ways than the existing ones?
Well, I mean, they will be different.
Okay.
So obviously, when one thinks about antimicrobials, the antimicrobials we have today in use, conceptually they're not that different from the penicillin that Fleming identified many years ago, right? In other words, the goal of the antimicrobials is essentially to kill, say, a bacterium without causing, obviously, harm to the person that is being treated. So that concept is the concept around which essentially drugs are generated today. And obviously bacteria are very versatile, and they're genetically very able to mutate out of a problem like facing the drug. So what I'm trying to say is that developing resistance to antimicrobials that put such a tremendous pressure on the bacterium, they either mutate or acquire some kind of information, or they die. There isn't too much of not too many options there. Okay. So the selection pressure, as it's called, is very strong, and therefore, obviously, antimicrobial resistance is a major problem these days, and there are pathogens against which we certainly have run out of drugs that we can essentially use. Now, looking at certain kinds of drugs of the future, and probably not the so distant future, these types of drugs would be aimed not necessarily at killing the pathogen, but rather to cripple it so that it can no longer cause disease. Now, for reasons that are a bit too technical to get into, the selection pressure in that case is less, and certainly resistance can always emerge, but less likely. There is another advantage of antimicrobials aimed at disrupting the ability of pathogens to cause disease, and that is one of the things that occurs when you apply a sort of a generic antimicrobial, that the ones we have today, is that they don't distinguish good bacteria from bad bacteria. In other words, you may know that we have something called the normal flora, for example, of your intestines, which increasingly we are becoming more and more and more aware of the tremendous importance that this flora has in terms of our normal physiology. So we can no longer afford to so casually wipe it out when we prescribe antibiotics. In some cases you don't have a choice, so of course you have an acute situation where you actually need to kill the pathogen, otherwise maybe someone's life is in jeopardy. Obviously we will still need those kinds of drugs. But there are other instances in which you may not need to act so acutely, and by simply crippling the ability of a given pathogen to cause disease, you may actually fix the problem. In other words, be able to treat the disease successfully. But those drugs, by definition, will not target normal microbial flora, because by definition they do not have these tools that pathogens have. So they are much more targeted drugs that will spare flora, and that will be an additional benefit that certainly we need to consider, particularly now that we're so aware of how important the flora is. So those are the kinds of types of drugs that emerge out of detailed understanding of how something so central to the ability of a pathogen to cause disease, for example, this syringe that I've been talking about, detailed understanding of the workings of that type of machine allows you to get at it in a sort of a more practical